Hello everyone. So today in this video we are going to talk about the Plasmodium species. So the Plasmodium comes under the Sporozoa, under the Protozoa. So the various species of the Plasmodium we can see. We can see the Plasmodium vivax, Plasmodium ovale, uh, Plasmodium falciparum, and Plasmodium malaria. So the disease caused by this Plasmodium species is malaria, and the main symptoms of this malarial disease is fever, headache, anemia, and splenomegaly. Now, what type of fever they manifest of different type of species? So, in the Plasmodium vivax and Plasmodium ovale, there is 48-hour cycles of the fever that we call as torsion fever. So, it includes fever on first and third day. So, fever actually is 48 hour apart, and the Plasmodium vivax and the Plasmodium ovale goes into the hepatic cycle, and they form a dormant form we call it hypnozoites that is found in the liver. So it can be reactivated in the later um, part of the life. So in Plasmodium falciparum we see the severe irregular fever patterns. The parasites RBC occludes the capillaries in the brain, and the symptom we see is of cerebral malaria. Also, the parasites RBC can occlude kidney and lungs, and it can damage those organs. The Plasmodium malaria has 72-hour cycles fever that we call as quartan fever. How this malarial parasite is transmitted from one person to another person? So main transmission occurs through female Anopheles mosquito. And how do we diagnose this? We diagnose it through the blood smear. We see a trophozoite ring that is formed within the RBC. We can also see the C zones containing the merozoites. We can see the red granules. We also call it the snuptional stippling throughout the RBC cytoplasm that is seen with the Plasmodium vivax and Plasmodium ovale. Now let us talk about the treatment. The treatment is mainly chloroquine if this chloroquine is sensitive, but if we find this chloroquine is resistant, then we can also get mefloquine or atovaquone or proguanil. So if the malaria is life threatening, we use the IV quinidine or artesunate for the Plasmodium vivax and ovale. We use or add primaquine for the hypnozoites, but before giving this primaquine we test the G6PD deficiency, because the primaquine hemolyze the RBC. So the person who has already G6PD deficiency, then there is more chance of hemolysis, and the person can suffer an anemic attack. So now let us discuss the detail about the life cycle of Plasmodium species. Now let us discuss the life cycle of Plasmodium species. So I have already told you that the Plasmodium vivax and the Plasmodium ovale goes the extrahepatic life cycle. They remains as the hypnozoite, and later on stage of life they reactivates on time and time. So we give a special anti-malarial drug that is primaquine for the Plasmodium vivax and the Plasmodium ovale. So let us start from the infective stage of the life cycle. That is the mosquito injects the sporozoites during the blood meal. So later on I will describe in this life cycle that after the rupture of the mature oocyst, the oocyst liberates the sporozoites, and the sporozoites reach the salivary gland of the mosquito. And when that infected mosquito is having the blood meal in our human body, then the mosquito injects the sporozoites during that blood meal, and that sporozoite is the infective stage of the Plasmodium species. So this sporozoite infects the liver cells. So you can see the sporozoite infects the liver cells, and there is multiplication of that sporozoites in the liver cell, and there is the formation of the C zones. You can see the many small rounded oval shaped structure, that is the formation of the C zones. So what happens is that there is rapid multiplication of the sporozoites in the liver, and there is formation of the many C zones, and after that there is liberation of the C zones by the rupture, by its rupture after the multiplication. So the multiplication happens so much that the liver cell cannot contain the C zones, and there is liberation of the C zones by its rupture. And after the rupture of that C zone, the merozoites are released from that C zones, and those merozoites goes into the bloodstream, and the merozoites that goes into the bloodstream invades the RBC by the invagination. So we have the sporozoite from the mosquito in the blood meal, and the sporozoite multiplied in the liver cell and it forms the C zone. And after the multiplication, there is liberation of the C zones and there is rupture of the C zone, and after that rupture of the C zones, the merozoites are released from the C zones, and those merozoites infects the RBC by invagination. So once the merozoites invades the RBC, you can see this is the RBC. After it invades the RBC, there is formation of the trophozoites. So there is formation of the early trophozoite and there is formation of the late trophozoites. The trophozoite is the amoeboid-like structure. So you can see the ring-like stage. You can see it is the signet ring-like structure. So if we make the blood smear during this stage, then you can see that this type of structure in the RBC, and it is the diagnostic phase. So I have said that the infective phase is the sporozoite stage and the diagnostic phase is the trophozoite stage. And this from the merozoites, when the merozoite invades RBC, I said it becomes early trophozoite and it becomes the late trophozoite, and the trophozoite stage is the diagnostic stage. After the late trophozoites, there is the formation of the C zones in the RBC, and in the C zone there is the rapid multiplication of the C zones, and after the rapid multiplication of the C zone, the RBC cannot contain C zones, and there is the rupture of the C zone, and from that C zone, from the RBC, the merozoites are released, you can see, and that merozoites can again invade the RBC and can go under the erythrocycle schizogony, or if the organism or the parasite is of Plasmodium vivax and the Plasmodium ovale, then it can goes into the liver cell and can form the hypnozoites, and or the merozoites from all the parasites goes into the liver cells, and it can again form the hepatic life cycle. So again there is formation, again there is infection in the liver cell, and there is formation of the C zones, there is liberation of the merozoite from the C zones, and they can again infect the RBC, and the erythrocytic life cycle and the hepatic life cycle can goes on and on and on until and unless we check this parasitic infection by the anti-malarial drug. And I have already said that those ruptured sporozoites, I mean when the C zones is ruptured from the RBC and the merozoites are released, the Plasmodium vivax and the Plasmodium ovale can infect the hepatic cells and can form the hypnozoites. So what happens is that from the merozoites, they can undergo the gammagony phase. So in the gammagony, two types of gammatocytes are formed. One is male gammatocyte, one is female gammatocyte. We call it macrogammatocyte and we call it microgammatocyte. The macrogammatocyte is of female and the microgammatocyte is of male. So the mosquito sucks the blood. When the mosquito is having the blood meal, they take up these gammatocytes and the sporogony starts in the mosquito. Now let us talk about the sporogony, when they take up this type of the gammatocytes from our body. So what happens is that in the gut of the mosquito, after taking the blood meal, there is exflagellation of the microgammatocytes, and that from the exflagellation, one of the flagellates go inside the macrogamete and form the zygote. And from that zygote, after the fertilization of the macro and microgametocyte, it forms the ookinete. So the ookinete penetrates the epithelial lining of the mosquito stomach wall and it forms the oocyst. So inside the oocyst, there is lots of sporozoites, and these sporozoites are multiplying rapidly. So upon the rapid multiplication, the mature oocyst ruptures and it liberates the various sporozoites. So these sporozoites reach the salivary gland of mosquito awaiting the another blood meal. And when this infected mosquito sucks our blood, then those mosquitoes inject the sporozoid during the another blood meal, and the life cycle goes on and on and on. So like this. So this is the life cycle of the Plasmodium species, the malarial parasite. So I hope you understood this topic on the Plasmodium species and its life cycle. So life cycle of the malarial parasite is one of the very important topic. So again, I want to emphasize on the infective phase and the diagnostic phase. The infective phase is the sporozoid phase, and the diagnostic phase is the early trophozoite stage and the formation of the C zones in the RBC. So this much for today in this video. Thank you.